So we talked about this, but biosimilars have to be systematically engineered to match the reference product. This is all reverse engineering. As you can see here, there's biosimilar and highly similar, and I'll come back to that. But you start out with structural and functional characterization, and that's a major portion of what you use to characterize biosimilars. The clinical trials and the immunogenicity are used basically to resolve residual uncertainty. So, excuse me. The challenge is, you know this would happen when we're late, is knowing which differences matter. And you can see we have aspirin, insulin, somatropin, and then we get to the IgG1 antibodies, and they're much more complicated. So we know already about primary, secondary, tertiary, and quaternary structure. And everything that EMA characterizes products by is quality, safety, and efficacy. And US FDA does it in terms of safety, purity, and potency. So what's more important is this reverse engineering leads to a huge chemistry, manufacturing, and control section because biosimilar's been engineered to match the reference product. And that's where all the data is. So then you look at the characterization, and will you think that the parameters of the biosimilar will match the reference product? In fact, the parameters for the biosimilar are much more narrow. They have much less lot variability, and they have much higher characterization because of the advances in technology over the last 20 years. So EMA has regulated biosimilars for a long time, since 2005, 2006, and they revised their guidelines. And then in 2012, they actually issued a new guideline specifically for monoclonal antibodies because there had not been a monoclonal antibody biosimilar prior to that time. And we talked about the Affordable Care Act and how it defines a biosimilar, so it has to have no clinically meaningful differences, same mechanism of action, same clinical result in any given patient. What's interesting is that the FDA has issued 5 draft guidance documents about biosimilars, and I've never seen one before on questions and answers. And they say that they're going to keep updating it as the questions get more interesting, or maybe as the answers get more interesting, I'm not sure. And I've never seen a guidance document on FDA meetings between sponsors and the agency. But that's also been issued, and that's because a PDUFA 5 is actually paying for the review of biosimilars. And finally, this past year, they issued one on clinical pharmacology data, and they have yet to issue one on interchangeability, which I will come back to in a minute. This is the type of fingerprint analysis of structure and ultimately function of monoclonals that we do. And you can see that it's extremely extensive, and you have it in your handout so you can look at it later. So essentially what we're looking at is when we look at an originator biologic under the new pathway of 351A, you can see there's characterization, non-clinical studies, phase 1, phase 2, and phase 3 makes up the biggest portion of development. We now turn that upside down for a biosimilar, where the structure and functional characteristics are first. There are abbreviated non-clinical studies, and then PK/PD, which also includes immunogenicity, and clinical studies are the smallest portion and used, again, as I said, to basically resolve residual uncertainty. So that's really the importance of those studies. Now, if we look at PK, we're talking about equivalency, no such thing as non-inferiority. So you're looking at Cmax, area under the curve, from first to last time point, and area under the curve from first time point to extrapolated to infinity. And all 3 of these geometric means must fully fall within the 90% confidence intervals from 80 to 125%, which is the predefined definition of equivalency that the FDA uses for antibiotics and for PK. What about efficacy studies? Well, at least one study has to be done because you have to have a pre-approval immunogenicity study and a post-approval immunogenicity study. And when you're looking at it, at least one and probably 2 clinical trials, One of them is going to be for equivalency to the reference product, and you need to preserve 50 to 75% of the effect size for non-inferiority, but you also want equivalence so that the lower limit of the 95% confidence interval doesn't cross the margin. Now, the definitions are based on previous trials, and I'll show you this with this Celltrion product, the Infliximab biosimilar. So the real differences between what EMA requires and FDA requires probably lie in extrapolation, if any. The only other thing is that the FDA requires one post-marketing immunogenicity study, which EMA does not do. So now this is a biosimilar from Celltrion that's marketed under several different names by two different companies. So it is marketed as Inflectra by Hospira, and that will be the name if it's approved in the US. It's also marketed as Remsima by Celltrion, which is a company in South Korea that makes this biosimilar. It's licensed to Hospira, and then they compete with each other in Europe in various countries, and I can't explain why they do that, but it's the same product with two different names. It's also called Flumigis in Hungary, where there's been about a 30% reduction in cost. It was one of the first places it was introduced as a biosimilar in Europe. And in Norway, because of negotiations with the government, Celltrion has provided the product at a 60% discount so that Norwegian government can sponsor a trial comparing switching to Remsima from Infliximab. So the two studies that were published in Annals of Rheumatic Disease that were used as a PK/PD study and then also ultimately as a efficacy study was one Planetus in ankylosing spondylitis and the other Planetra or Planet RA in rheumatoid arthritis. So this was the first study, and this was the second study. Essentially, the EMA recommended approval in June of 2013, and essentially marketing approval after that. And they can only recommend it, so subsequently it gets accepted by the member countries. They extrapolated data to all of the Remicade indications, and of course they set up a pharmacovigilance plan, and it has the same label as Remicade. Health Canada approved this product, but not with extrapolation to inflammatory bowel disease, only to psoriasis and psoriatic arthritis. Now, if we look at Inflectra and Remicade, they're actually very, very similar. It has to have the same primary structure, and that's a rule by the biosimilar requirements in the US. The tertiary structures are also identical, and there's comparable folding of the proteins. There was some small differences in fucosylation or absence of fucosylation in the FC portion of the molecule, which did not change PK/PD, did not change the stability, and did not make a difference on immunogenicity. And then when we look at the mechanism of action, we can see that TNF neutralization, binding affinity, all of these were quite comparable. The only difference between the Celltrion product and Remicade was relative binding to the Fc gamma 3A receptor in those patients who had a V or F polymorphism, where then there was a slightly less binding. Now, we know already that those patients with those polymorphisms have been studied specifically in the ACCENT trial in Crohn's disease and ATTRACT in RA and show not to have a difference in response. And we actually believe strongly that the mechanism of action of all the TNF inhibitors is not mediated by ADCC or CDC, and I'll come back to that. So if we look at the extrapolation summary, it was very extensively characterized from biologic activity, neutralization, and they showed a lower amount of afucosylation, as I mentioned, but in fact, EMA did not think this would have a clinically relevant impact, and thus they extrapolated. And when you look at the analyses under physiologic conditions, in fact, when you use whole blood and you use whole blood derived from IBD patients and use lamina propria cells, then you find that there really is not a difference between these products. They do not behave differently ex vivo in an IBD model. Now, we still really don't know how well TNF inhibitors work or exactly what their mechanism of action is because it difficult to sample at the site of inflammation. We've always used in vitro assays with very high levels of TNF-alpha, but we do believe now that binding and neutralization of soluble and transmembrane TNF are the two mechanisms, and in IBD, it's presumably more transmembrane TNF because that leads to reverse signaling and apoptosis through cytokine starvation. But that mechanism also applies in RA. And what we do know is that with certolizumab pegol, without an FC receptor, it cannot possibly mediate ADCC or FC, and in fact it's quite effective in Crohn's disease. But it was never reviewed or approved in Canada, so that's why probably the Canadian authorities decided not to extrapolate. We also know that etanercept does not mediate binding to transmembrane TNF very well. So when we look at the open label extensions from Planetra and Planetas, we see that there were no real apparent differences, but they only did a single switch. And so that's not enough for us to know exactly what interchangeability means. And what we now know is that FDA has announced that they would like to have comments on interchangeability, but that they say that the review time for interchangeability will be comparable to a brand new BLA. And what that means is that they're implying they're going to expect a series of randomized controlled trials like they required for abatacept going from IV to sub-Q, tocilizumab going from IV to sub-Q, which is like about 2,000 patients and up to 4.5 years of treatment, repetitive switches back and forth, and discontinuation, and then restarts. And so I do not think at any time soon we will have interchangeability because that's already now the cost of a new development program, and it may be better to just develop a better biologic. Interchangeability has been defined lots of different ways, but it's only the FDA that really has a level of interchangeability, and it's higher than just being a biosimilar. Now, substitution is something that the pharmacist can do. But the rule is that unless the FDA has defined a biosimilar as interchangeable, there will be no substitution without the healthcare provider's knowledge. And I'm here to say to you that I don't think we're going to see an interchangeable biosimilar anytime soon. So despite all the concerns in the state legislatures, I think we will have to be reassured that even if we're going to be forced to use the biosimilar, we will know that we are being forced to use it, and the patient will know that they're being forced to use it. So in conclusion, I didn't show you the clinical data because of time, but you have it, I believe, in the slide set. The PK/PD was essentially identical. It overlapped in the Planetos or AS study. The efficacy was highly similar within a very tight level of equivalence of a 13% margin on either side based on the ATTRACT data. And they used the lower dose of 3 milligrams per kilogram with methotrexate in RA because that would show more immunogenicity. And they used a dose of 5 milligrams per kilogram with no background therapy and ankylosing spondylitis to again mimic what was done with the reference product. Now, we know that there are a lot of biosimilars in development, and the only thing I can tell you is that the FDA postponed the meeting in March to review the Celltrion biosimilar product, and we have not heard when it will be rescheduled, so that will be of great interest, and we're all waiting to find out. So thank you very much.